So the next speaker is Thomas Hiron from the RVC, and we're looking at novel transcriptomics, insights into host immune repertoire, intra-host virus mutation in Feline Infectious Peridonitis. Thank you so much.
Hi everyone, I'm Tom. I'm a postdoc in Lucy Davison's lab, hosted jointly at the RVC and also at the University of Oxford. And today I'm going to share some of our work on coronavirus-mediated diseases in companion animals. So specifically, I'll be talking about what we've done on Feline Infectious Peridonitis, or FIP. Fortunately for me, you've already had a bit of an introduction to FIP and feline coronavirus this morning, so I'm actually going to skip over most of the introductory stuff on this slide. The reasons that we're interested in FIP, the pathogenesis is very much not fully understood. Host and viral factors have been associated, but there's no clear understanding of why some cats, when infected with feline coronavirus, will go on to develop FIP, whereas others will recover fully. Currently, there's only a single vaccine available, but its use is actually advised against due to low efficacy and a possible increased risk of FIP in vaccinated cats through antibody-dependent enhancement. And certainly in the past few years, antiviral therapies have shown a lot of promise in ameliorating symptoms in cats with FIP. However, there are reports emerging that recovered cats may continue to shed virus, raising concern around development of antiviral resistance. So in 2020, a few years ago now, we started our project, which we named Mapping Animal Susceptibility to Coronaviruses, Outcomes in Transcriptomics, trying to cram that into mascot for short. And this was built around a feline tissue sample archive maintained by Dr. Emmy Barker at Bristol Vet School. And the archive comprises multiple feline tissues from cats euthanized for a variety of reasons, including FIP. So we selected samples from this archive, and then there were three major arms to our study. The first of which was whole genome sequencing in a limited cohort of cats with and without FIP. And this was to identify candidate FIP-associated genetic variants. This was followed by successive rounds of targeted validation sequencing and has resulted in a list of FIP-associated genetic variants to be published very soon. The second arm was an attempt to better understand the feline MHC, which is quite poorly resolved in current feline genome assemblies. And to do this, we created a new whole feline genome assembly using nanopore long-read sequencing, and followed this up with a targeted nanopore-based approach to directly sequence the MHC in a number of FIP and control cats and investigate potential association with FIP. But today I'm going to mainly focus on the RNA sequencing arm of our study, where this led us, and just a bit of what we found is quite a lot of data, so I'm going to try and whisk through it in a sort of coherent way. And just to note that there is a manuscript very near completion describing these results, so some of the results have been sort of anonymized. So in terms of the host transcriptome, we sequenced RNA from 4 tissues from cats with and without FIP. These were the kidney, liver, lung, and mesenteric lymph node. And when we looked at the overlap between tissues in terms of genes with higher expression in FIP, we weren't really surprised to find that most of them were interferon-stimulated genes or somehow otherwise involved in type 1 interferon signaling. In fact, the 24 genes that were significantly more highly expressed in FIP across all 4 tissues were all interferon-stimulated genes and showed a similar degree of high-level expression in cats with FIP. We also observed higher expression of genes involved in B and T cell mediated immunity, more generally the adaptive immune response, most prominently in the mesenteric lymph node samples that we sequenced, but also evidence for that in other tissues as well. And when we looked at how some of these genes were expressed among our cats with FIP, we found that while interferon-stimulated genes were quite evenly expressed, with the exception of the 2 cats shown on the heat maps with a more focal renal form of disease. Fairly even expression of interferon-stimulated genes, but the expression of other genes such as those encoding inflammatory cytokines and chemokines, and also genes involved in the adaptive immune response, were more heterogeneously expressed. These are the mesenteric lymph node samples as an example. And using information we had from immunohistochemistry, we were able to see that this appeared to be correlated with the abundance of virus in those tissue samples. We then wondered if we could directly measure abundance of the virus from our RNA-seq samples by mapping the reads onto a feline coronavirus genome, which we were able to do. And when we looked across all the tissues in our data set, we found that this correlation held up and did appear to be related to viral load in the samples, which suggested to us that, at least in some cats, feline coronavirus is able to persist despite very high levels of inflammatory gene expression and immune activation. So as a brief aside, we were able to assemble full-length feline coronavirus genomes from those reads that we detected in the samples in all tissues except for the kidney. And when we compared those to some public feline coronavirus sequences, we found that they clustered with serotype 1 feline coronavirus, which was expected as the most prevalent serotype in natural infections. And we also noted that there was no clear phylogenetic signal segregating the pathogenic so-called FIP virus isolates, including our assemblies, from the non-pathogenic feline enteric coronavirus sequences, which is consistent with previous studies. But the upshot of this work is that we can actually, by mapping the RNA-seq reads back onto our feline coronavirus genome assemblies, we're able to measure the frequency of single nucleotide variants across the length of the feline coronavirus genome. And because we've captured and sequenced multiple tissues from the same cat, we can actually track the frequency of those variants across tissues and infer sites under selection within each cat. But unfortunately, I don't have too much time to talk more about that today. The last part of this project that I want to talk about is our analysis of the immune repertoire in FIP. And by immune repertoire, I mean the collection of T and B cell receptors expressed within a cat that determine its adaptive immune response to infection. And this major oversimplification of VDJ recombination, which determines this diversity and specificity, is really just to illustrate the fact that we can directly capture this information from the recombined receptor sequences as expressed as messenger RNA. And then this is an illustration of just where we've done this for the immunoglobulin heavy chain locus, showing that when we break down the immune repertoire into categories based on how abundant those clones are, how expanded they are, we can see that hyperexpanded clonotypes, shown in blue, in the dark blue over here, are more commonly found in samples from cats with FIP compared to controls. So we've extended this analysis to a follow-up study in a much larger number of samples using a sort of PCR-based library preparation method to specifically enrich for TCR and BCR sequences. And here I'm going to show you a specific example where we found an association for the alpha chain of the TCR that seems to distinguish FIP in control samples. So when we take the sequences that we've obtained, we can produce these immune repertoire networks for each sample, which can give us a kind of visual representation of the specific receptor sequences that are present. And using this information, we can then use network-level statistics to infer differences. So here I'm just showing you an example using the Gini coefficient, which is a measure of evenness, so how evenly the reads that we've sequenced are distributed among clones, represented by the nodes in the network. And there appears to be no significant difference between all FIP and all control samples in our follow-up cohort. But when we, again, use the information we have from IHC, it does appear that in samples that have shown to be positive for the virus by IHC, there is a significant increase in the Gini coefficient, which suggests that there is, it's more uneven in terms of how those reads are distributed among the clones. So the inference then is that actually there are more of these expanded clonotypes appearing in FIP samples compared to controls. And we can dig a kind of level deeper and look at the CDR3 receptor sequences that are associated with either FIP or control cats. And this is just a heat map showing you the number of clonotypes for each clustered sequence that occur within our FIP and control cats. And we can see that the majority are associated with FIP samples, but actually no single receptor sequence or cluster is very strongly associated. However, when we look in the other direction, our top hit was a TCR alpha sequence that was found in almost 80% of our controls, but actually very rare in our FIP samples. So at the moment, we're quite curious to know whether those are associated with protection from FIP and pending some kind of functional follow-up, which we hope to look at in the very near future. So just to summarize then, we found that RNA-seq RNA-seq reveals a systemic activation of type 1 interferon signaling in cats with FIP, affecting multiple tissues. And this appears to be correlated with abundance of feline coronavirus in those tissue samples. We can recover full-length feline coronavirus genomes from our tissue RNA-seq, and this enables analysis of intra-host single nucleotide variants. And lastly, the immune repertoire does seem to be altered in FIP, and we've begun characterizing those clonotypes, picking them out, and seeing exactly how they're associated with either presence or absence of disease. So just a quick advert that we, as well as a manuscript describing these results, we have another couple in preparation, one describing the host genetic susceptibility part of the study that I mentioned earlier, and another describing the work on the feline MHC. And we've also recently secured additional follow-up funding to advance some of this work, in particular looking at the FIP-associated genetic variants and functionally characterize those in vitro. So all that remains is to thank our many collaborators, some of whom are here today, and of course our funders, predominantly UK-ICN, who originally funded the project, and PPCT, who continue to fund the project. I don't know how I've done for time, but I'm happy to take any questions.
Very well. Thank you so much for sticking to the time. That's also great. If we have a question, yeah. In the meantime, I'll squeeze mine.
Sorry?
In the meantime, I'll squeeze my question
Sure.
when we reach that. I was looking at the TCR repertoire, and I was wondering if you find a difference in the differential disease. So if it's a granulomatosis form or exudative, or you find different?
Yeah, so it's quite, so you mean in terms of wet and dry FIP? Yeah, so the challenge is that although some cases are quite easy to characterize, many of them have features of both. So we actually shied away from putting a kind of broad wet and dry FIP characterization. But certainly in some cases there'll be more specific, say like neurological signs. And that's not something we've looked at in granular detail yet, as we're just pulling apart the kind of global associations, but it's definitely worth looking at, I think.
Yeah. Thank you. Dara?
So this is coming from a non-expert. So it's been unclear to me mechanistically underpins the transition from COV to FIPV, whether or not that's viral or host. Sorry. No, that's good. It was interesting you have the viral sequence. I think it was temporal. So I'm just wondering if it. I've heard discussion about loss of the therapeutic supply, or is it that? Or is there a more complicated story?
Yeah, I think on your first point, it's probably unclear to a lot of people in the FIP space as well exactly what's going on. I mean, various mutations have been associated with FIP in the past. I think more recently it's become clear that actually they're more likely to be associated with systemic spread of the virus, as cats have been found with systemic FCOV infection, but actually no signs of FIP. So it's still unclear. We believe that there's very much a host component at play. And we hope that, potentially some of our immune repertoire work, for example, would show, why some cats may respond, in a good way to the virus infection, whereas others are unable to clear the pathogen and result in FIP. But yeah, it's not known really.
I'm not there.
Great talk. I'm not an immunologist, so this might be a really easy answer. So my question conclusion is not working.
It's
really close. Okay. Is the conclusion that antibodies are diversifying but not producing any effect to clear the virus because of lack of innate immune response? Or is it
possibly
more complicated?
What's known is that in cats with FIP you kind of get hyperglobulinemia, so you get massive increased production of antibody, but actually it may not be beneficial. And in fact, I mentioned antibody-dependent enhancement. There's some quite old evidence that may be a feature of the disease itself. So the kind of general idea or opinion out there is that B-cell mediated immunity is probably not beneficial, whereas the cell-mediated immunity through T cells may be more beneficial for clearing the virus. And in terms of the innate immune response, because we've pulled samples, these are post-mortem tissue samples from cats with and without FIP. They're going to be at different disease stages. You know, I've always wondered if, like you see in COVID-19 with the strength of the initial interferon response, you know, is that playing a role in FIP? We don't know, but it would be useful to get hold of, it would be interesting to get hold of some samples and investigate that, you know, the role of type 3 interferon signaling, for example. But yeah.
We're going to be short of time. If there is one last question there, yeah.
Just wondered, in different tissues, were there any differences in viral sequence as well?
Yeah, so actually the virus genomic sequence was quite similar between different tissues, but that's kind of what I was hoping to highlight with that plot that I shared for about all of 10 seconds. Actually, there are certain variants that do change in frequency. We kind of used the mesenteric lymph node as a putative first site of infection from the gut as the virus moves around the cat's body. And it does seem that some of those variants end up fixed in other tissues. So they aren't numerous. The genomes are still quite similar in terms of RNA sequence identity. But there certainly are sites that seem to change, and maybe even the same sites in different cats. So.
Thank you very much.
Cool.
It was really great.